Welcome back, students. We are moving on now to chapter 2 of the Pinel book, Brain and Behavior. And naturally, we're going to get into more and more detail about the brain. It's very important to us. The ancient Greeks used to believe that they didn't know what the brain did. They thought it was a radiator, and that that our body heat was radiated from our brain. I guess that sort of made sense in a way. But anyway, we are moving on. This is a long section. I've got about 54 slides here, and they're all packed with some detailed information. I might have to split this into two parts. We'll see as we get further into this lecture. So, moving on. Let's talk about what neurons are. A neuron is an individual nerve cell. We have about 100 billion neurons, just 100 billion neurons in our brain. That's not including all of the interconnections. Some neurons can make hundreds, even thousands of interconnections. There are literally more and I mean literally more interconnections, neuron interconnections in our brain than there are stars in the universe. Now, glial cells are cells that are also in the brain. They're connected very much to neurons, but they are support cells in a variety of different ways. We'll talk about those later, about glial cells later. But neurons do interconnect into very vast networks, and those networks give rise to intelligence and consciousness. The parts of a neuron, the very basic parts of a neuron, the dendrites. Here you can see the dendrites as my cursor is moving around, and this entire structure is the cell, the nerve cell, the neuron, the dendrites, and they receive messages from other neurons. So you can see all these various branches out here where other neurons interconnect. The soma is the cell body. It's the body of the neuron, and it's this part right in here, the part that the dendrites are streaming out of. The axon is a fiber that carries information away from the cell body, and it goes out here. There's the axon. But now you'll see at the axon also that we have other cells just simply around this, the length of the axon. They're called Schwann cells. And they create a sheath around this, or around the neuron, called the myelin sheath, around the axon, that is. And then there's spaces in between the Schwann cells, and those are called nodes of Ranvier. So we have the axon, and that's a fiber that carries the information away from the cell. The axon terminals, and they're out here, and they are the branches that link now the dendrites of other cells into the neuron. So here's just a graph of different, not graph, but a graphic of different types of neurons. Here's a unipolar neuron, and you can see here unipolar. So we've got out here dendrites out here on these sides, and these are the axons. The axon is this long length, and then the axon buttons or terminals out here. So here's a unipolar cell, a bipolar cell, a multipolar neuron, and these multipolar neurons, as you can see, like this one here, is just simply a cell body with a bunch of dendrites coming out. Now, the nerve impulse itself, the basic so-called nerve impulse, is an electrical message, and it's transmitted down the length of the axon. similar to how a wire works, but it really is not. So when we talk about how a signal or a neural message travels down the axon, indeed, it's sort of like a wire. But in fact, the signal starts out up here in an area just associated right at the very end of the cell body, the soma, between the soma and the axon. It's called the axon hillock. It starts back in here. And if the signal gets a certain threshold, it's sent. I mean, if the power here or the energy reaches a certain threshold, a signal is sent down. We see a signal that goes down the length of the axon, and you can see it just goes basically from node of Ranvier to node of Ranvier, straight down the axon, and then we'll see what happens when it gets out to the axon buttons. But the speed of the neuron, the speed of the axon itself, or the pulse itself, can go anywhere from 1 meter per second to 100 meters per second. We usually don't think of impulses, neural impulses, as being quite that fast, but indeed they are. And as you can see on your screen here, the action potential is that nerve impulse that's triggered when the neuron reaches its threshold or triggering point for firing. So it's as soon as it gets to that triggering point, as soon as it gets above that threshold, boom, you have that action potential fire. Now the action potentials aren't variable. It's either it is either a specific action potential or it doesn't fire at all. The reason it can fire is because of little tiny openings in the axon membrane. They're called ion channels. And when the axon is carrying a pulse down its length, as you can see on the graphic here, as that action potential proceeds down the length of the axon. At the same time, very, very fast, these ion channels are opening and closing and opening and closing and allowing potassium ions and sodium ions to interchange back and forth across the channel. And hence, because of that interchange, that pulse travels down the length of the axon. So during the action potential, you can see the ion channels opening, and they allow the positive ions to rush in, and very briefly pushing the electrical charge up to 30 millivolts. Here's another graphic, and it shows you a little more detail of the action potential. So the ion channels, they open up near the soma, and as I said before, that area where it opens up, and I'll show you that later, that area where it opens up is called the axon hillock. And then gate after gate opens up straight down the length of the axon as the action potential zips there along. If you've ever seen a domino cascade, somebody lines up a bunch of dominoes and then tips one over and the rest of them fall like that, it's the same thing. It's the same idea. As soon as one domino falls, then you get the stream of dominoes falling all the way to the very end. And that's what happens in the axon. You have the sodium and potassium ions going across the channel in those gates, and you can see that the sodium entry, it what we call depolarizes the membrane, and then that opens up additional channels. Saltatory conduction is a process where nerve impulses are conducted down the axons, and it's coated with and those axons are coated with a myelin sheath. But like I said earlier, and as you can see here, the myelin sheath is segmented. There's segments, and they are segmented segmented, that is, Schwann cell per Schwann cell. So it's not like a wire, or like an insulated wire where you have just some current going down a wire. Plug it in and turn it on. It's not quite like that. In fact, it's not like that at all. Many times you'll hear somebody use a metaphor or liken an axon to a wire, and how the action potential flows down the axon. But that's not really how it works. And the myelin sheath in itself isn't an insulator like you see insulator on a wire. But without that myelin sheath, without that fatty layer coating on many axons, some don't, and those axons that don't have a myelin sheath are much, much slower. So the myelin sheath greatly aids in the conduction or speed of the action potential down the length of the axon. Now, a synapse is a microscopic space between two neurons. And in fact, the synapse is a space between the axon terminals and the dendrites of an adjacent neuron. It is that synapse across which one neuron communicates with the other neuron. They're not tied together physically, spliced together like two wires might be spliced together. It's not like that at all. A neurotransmitter is a chemical that alters activity when they cross the synaptic gap, that little space between two neurons, between the axon terminal button and the dendrite of the adjacent axon. It's that gap. And then they will either excite or inhibit. They'll either send an excitatory or an inhibitory signal to the next neuron. So some neurotransmitters excite, and by excitation I mean it moves that neuron closer to firing, and some inhibit, so it moves it away from firing. A receptor site is the area on the surface of a neuron that's sensitive to neurotransmitters and also hormones, which sometimes serve as neurotransmitters or a type of neurotransmitter. So here's a neurotransmitter. You can see the synaptic cleft here. This is the synapse right in here. Here's the axon. Here the axon's coming down, and this ends up in this terminal, this little, we call it a little button, a terminal button. Vesicles here are little reservoirs filled with neurotransmitter molecules. So the vesicles are stored up back here. They're little reservoirs, and when they come out here to the end of the axon, they come out here and erupt through the membrane into the open area, the synapse, and then there's receptors on the other neuron, as you can see here. So there's one neuron here, there's the axon, here's the neurotransmitters, and this is the dendritic side of the adjacent neuron. Right here we have receptors. Now, a lot can happen in here, and in fact, what happens in here is probably 50%, if not more, of the main story of many pharmaceutical companies, especially companies that make pharmaceuticals for psychological issues of any order, anything from schizophrenia to depression to anxiety to ADHD to PTSD. The area that is most targeted are synapses of axons. Now, of course, it depends upon where in one's body or where in one's brain is relevant to the particular action involved, but that's really where it falls down. That's about where it falls, is in the synapse, and how one neuron can affect another neuron, and so on down the line. What are some of the major neurotransmitters? What are their names? You may have heard of some of these. One is called acetylcholine, and it's an excitatory neurotransmitter. It is mostly involved with like motor function, movement, what we call autonomic function. We'll get into what that means in a few minutes. It also participates in learning and memory. You've probably heard of dopamine. Dopamine is also an excitatory neurotransmitter, and it's in motivation and reward. It's responsible for making you feel good, and lack thereof of dopamine can bring you down. GABA, GABA, is an acronym, and it stands for let me see if I remember, gamma aminobutyric acid. GABA is gamma aminobutyric acid. It's an inhibitory neurotransmitter, so that means that it exerts inhibition. It modulates the central nervous system. And it participates also in mood. If alcohol, for example, is responsible for inhibiting GABA. So therefore, if you inhibit an inhibitory neurotransmitter, that means that things are freed up more. And and that frees up everything, not only with thinking and emotion, but also with motor, with your motor system and executive operations as well. Another neurotransmitter is called glutamate, and a lot of the research just recently, in fact, in the past 10, 15 years, has discovered that glutamate has a huge plays a huge role in memory. So it is an excitatory neurotransmitter as well, and it exerts excitatory control in our central nervous system. It participates in learning and memory, but to even a greater extent than we had known before. Then there's another neurotransmitter called norepinephrine. It is an excitatory neurotransmitter. Serotonin, which you may have heard of as well, is also an excitatory neurotransmitter, and it's also participates in various parts of the brain for mood and appetite and sleep. And again, all the pharma companies will focus on these various neurotransmitters and the effects that they have in the synapses of the neurons in whatever part of the brain is most affected or appropriate. Okay, now let's talk about also neural regulators. Neuropeptides. Now, anytime you see the word peptide, peptide is a protein. It's a much bigger molecule. Neuropeptides regulate activities of other neurons, so they're like controlling, making sure that the other neurons don't go haywire, more or less. Here are some of the different types of neuropeptides. So enkephalins relieve pain and stress, and they're similar to endorphins. We'll talk about it. So endorphins, this is released by the pituitary gland, and they also help to relieve pain. In fact, the opiates and opioids and morphine, particularly the molecular configuration, how the molecule looks, is very, very similar to endorphins. So in fact, most of the analgesic drugs, the stronger analgesic drugs, analgesic means pain reliever. Like when you go into CVS or something, and you look around and you want a pain reliever, instead you might see a sign over the aisle and it says analgesic. That means pain relievers. Endorphins are, in fact, strong pain relievers. A lot of you'll hear if you run, people talk about the runner's high. The runner's high, in fact, is the body releasing endorphins, which are very similar looking to morphine, and that's the runner's high. Surprise, surprise. All right. Then we have what are called neural networks, and naturally, with 100 billion neurons and 100 times more interconnections, those neurons synapse with one another and they form complex, vast neural networks. And that's what we are. That's what each one of us are. It's this vast number of neural networks that is a composite of everything that we've experienced, everything that we've learned, everything that we've reacted to, even things that you didn't react to. It's part of that makes each individual what she and he is. Individual neurons combine excitatory and inhibitory information from input neurons, and based on that, there's a sort of a decision to fire. So if there's, let's say that a neuron is getting a lot of inhibition neurons or signals, but also excitatory signals, if the excitatory signals exceed the inhibition signals, then the neuron will fire, and inversely, if the inhibitory signals exceed the excitatory signals, then the neuron will not fire. Neural networks also, remember, are neuroplastic, and that they allow our brains to change in response to experience, any experience. Every single microsecond that you're alive, you're experiencing, and therefore you are changing. So don't say you don't change, because you're changing right now. Just by listening to this, you're changing. Just by sitting where you are, standing, or whatever you're doing, you're changing. All right. We're going to talk about now we're going to categorize our nervous system. Our nervous system, that is all the neurons in your body. We're going to split it, we're going to say in two parts. One part we call the central nervous system, or CNS, and it is your brain and your spinal cord. That's it, period. The other part is called your peripheral nervous system. Now, your peripheral nervous system then has two parts. And remember, I mentioned the autonomic nervous system earlier, and I'd get to that. Now we're going to get to it. So your peripheral nervous system is composed of your autonomic nervous system and your somatic nervous system. Your autonomic nervous system communicates with your internal organs and glands. It will arouse you or calm you. When you're scared, your heart goes up, and you feel, your eyes, your pupils dilate, and you're scared. If you're aroused, then what happens? You constrict, you lose your appetite. Well, those are all functions of either your parasympathetic or your sympathetic nervous system. So your sympathetic, parasympathetic nervous system work with one another in concert. And those are part of your autonomic nervous system, along with the associated glands as well, and we'll get to those. Your somatic nervous system communicates with your sense organs and your voluntary muscles. So your sensory system, and we call that the afferent system. The afferent system is sensory input. So if you touch something hot, boom, it comes. It's cold. All sensory input is afferent, and that's part of your peripheral nervous system. Your motor system is efferent. So afferent is everything coming from outside into your brain. Afferent. It's first. Think of A. So that's first. The efferent or motor system, E, goes out. So your brain processes that, or your spinal cord may say, hey, no time to process that brain. We need to move that hand from the fire. In which case, the brain doesn't even see it first. You just go straight to your spinal cord and right back out into your muscles, and boom, you move your hand real quick. And then you may think that you realize it at exactly the same time, but you don't. There's couple milliseconds there until you actually realize that you burned your hand. Now, what's the difference between a nerve and a neuron? Neurons are single nerve cells. okay? A single nerve cell. Nerves, like you're getting on my nerves. Well, nerves are large bundles of many neuron axons. A neural lemma is a thin layer of cells wrapped around axons outside the brain and spinal cord. They form sort of a tunnel around damaged fibers and allow them to repair themselves. But the central issue here is that it's to understand the difference between a nerve and a neuron. The peripheral nervous system, as I said, composed of two parts. The somatic system, which carries messages to and from skeletal muscles and sense organs. It controls our voluntary behavior. If you think of moving your hand or whatever you're doing, that's voluntary behavior. Those are your muscles. That's a peripheral nervous system. The autonomic nervous system, or ANS, it works without you thinking about it. It's your internal organs and glands. It controls your automatic functions, such as your heart rate, your digestion, your blood pressure, your sweat, whether or not your hair stands on your skin stands on end or not, if you're scared or if you're cold. That's the autonomic nervous system. Okay, so this little diagram with all of the rather gory anatomical parts stretched out here tell you the parts of our bodies that the parasympathetic division of the autonomic nervous system are control, and the sympathetic division. So the parasympathetic division constricts pupils, the sympathetic division dilates pupils. And then go on down the line here, and you can see some of the various ways in which our autonomic nervous system is affected by, or that is how the parts of our body and how we feel and think are in fact affected by our autonomic nervous system. Now, the spinal cord itself connects our brain to all the other parts of our bodies. There are 31 pairs of spinal nerves, and they carry sensory and motor messages between the spinal cord and the body. 31 pairs of nerves, 31 pairs, okay? So let's talk about the cranial nerves first. The cranial nerves are 12 pairs of nerves that leave the brain directly, and they also work to communicate messages between the brain and the body. So there are spinal nerves and cranial nerves. 31 pairs of spinal nerves, 12 pairs of cranial nerves. Notice here also that the spine itself is separated into four parts. The cervical vertebrae here and here, then the thoracic vertebrae, the lumbar vertebrae, and the sacral vertebrae. All of these very important as we go down the spine, down the trunk. Of course, the efferent and afferent nerves going into and out of the spine also are controlling lower and lower and lower and lower parts of one's body. Be aware that these cervical nerves up here, C1 through C8, the C1 through C8 nerves, if especially above C5, if there is a fracture, if your spine is broken, above, as I'm sure you're all aware, if your spine is broken anywhere along here, you're going to lose that function in both sensory ability. You won't be able to feel whatever your spine serves below, wherever the break was. You won't be able to sense. You won't be able to move. Now up here, well, this lower brain part, this lower brain section where your spine is connected to your brain, that regulates heart rate, blood pressure, respiration, even body temperature. So if there's a fracture, an instantaneous injury or fracture up there, that almost always I won't say almost, it always guarantees death. Ideally, that is how hanging is supposed to work. So when the hangman makes the knot around the executed one who's being executed neck, and then pulls that lever and the person drops. They're also supposed to have a weight on the bottom of the person's feet so that when they drop, it snaps their cervical vertebrae, and the death is instantaneous. Now, of course, you see, especially in cowboy movies, they hang somebody over a tree or a limb or something. That's not really hanging. That's suffocation by hanging from a rope. So the spinal cord and behavior, the reflex arc, that's the simplest behavior. If you touch something painful or hot, just snatch your hand away or whatever. A sensory neuron is a nerve cell that carries messages from senses to your central nervous system. A connector neuron is a nerve cell that links to others, and a motor neuron is a nerve cell that carries commands from our central nervous system to muscles or glands. The production of new brain cells to replace ones that have been lost is called neurogenesis. And as we get older, the process becomes less and less rigorous. So we first have neural stem cells. So neural stem cells form. Some stem cells can become glial cells or support cells. Half of the neural stem cells actually that are formed die off. And then only a small percentage of the neural stem cells really become neurons. In the past 40 years, we've become more and more sophisticated at looking at our brain, at imaging our brain, and knowing what a brain does. And believe me, if it seems complicated at this point, wait another 10, 20, 30, 40 years, it's going to get even more so. We are at the point now where we can, in some cases, watch a thought proceed. And I'll explain what I mean by that later. But let's talk about some of the ways in which we can look at how a brain is working, how our brain is functioning. The first is called a CT scan. We all call them a CAT scan. It stands for a computed tomographic scan, and it is simply just a computer-enhanced X-ray of the brain or body at different angles. Now, an MRI stands for magnetic resonance imaging, and it uses a very strong magnetic field, not an X-ray, okay? Not an X-ray, it's a magnetic field to produce a 3D image of the body's interior. Now, the difference between a CT scan or CAT scan and an MRI is that the X-ray goes right through soft tissue. And it will only bounce off of harder tissue, like bone. But the MRI gets it all. What do we mean by localization of function? It is a research strategy of linking specific structures in the brain with specific psychological or behavioral functions. So if we can link specific structures in the brain, and we can't look at the brain and go, oh, that moved and therefore that moved. So we have to have experimental methods and techniques by which we can watch a change in the brain and a concomitant change outside. So how do we explore brain function? One is via and this has been the case for centuries, is the clinical case study. And these are just individual cases of people who suffered brain damage, and then we study them, we examine and do research, and we're going to get into this quite a bit as well. People who have had brain injuries of different types, we've learned a tremendous amount from these people because we know, and they knew how they were. And then through research with them and talking with them and through their cooperation, of which many have been marvelously cooperative and have helped science out quite a bit, we know the changes, and because of that, we know that certain structures were injured, certain changes then developed. So that is the first place in which we have gained information about brain function and localization. There's something else we have, what's called neurological soft signs, and they're telltale signs that a person's brain isn't working quite properly, like an awkward gait, or maybe poor hand-eye coordination, or if you go further into psychology, let's say that you give somebody an intelligence test or some other kind of test, and you see that there's, where if let's say you've got 10 different batteries in the test, and 5 of them for one thing, and 5 of them for the other, and you see that they should pretty much all be equal, but you see there's a deficit in one area. That's also a signal that there's probably some neurological soft sign going on. We can also learn a lot about the brain and through electrical stimulation. So we can put an electrode and activate target areas in the brain. And I'm not just talking about human brains. In fact, far more, we're talking about non-human brains. We can activate a certain activated target area in the brain, and then through that activation, we see a change, a resultant change. We can also do ablation, or these are surgical removal of parts of the brain. Now, and this includes humans as well. Let me point out something we'll talk about also, called a prefrontal lobotomy that used to be done. It's not done anymore. But again, 40 or 50,000 people, I think, had been subjected to this, to prefrontal lobotomies. Then we also have what's called deep lesioning, and it's this where we have an electrode lowered into a very specific area inside the brain, and then provide a stronger electrical current in there just to destroy a small part of the brain tissue that's there. This technique has been used not only for research in non-human subjects, but also in human subjects. For example, if a small part of the brain is causing seizures, we'll go in there and just deep lesion that very small part. We can also explore brain function using what's called electroencephalography, or an EEG, brain waves, in other words, and that detects, amplifies, and records electrical activity in the brain. Then there's something else that's fairly in the past 20, 30 years, called positron emission tomography, or PET scans. And these are computer-generated color images of brain activity. These work somewhat differently than an MRI, and that what these sense is that an individual first has to ingest a radioactive glucose. Now, it makes that just because that sounds worse than it really is. Radioactive doesn't mean that they're going to start to glow. It's very, 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 very light, and I mean it's less radioactive than an X-ray even. But in positron emission tomography, that glucose is a simple sugar, and it is basically the essential fuel by which we run. That's how we get our energy. So the brain, which consumes about 50% of our body's energy anyway, using those glucose, those little radioactive glucose molecules, positron emission tomography can tell exactly what part of the brain is using more energy, therefore using more glucose, than other parts of the brain. So that's a PET scan, and then we have also what's called a functional MRI. We had a plain old MRI. Now, a functional MRI actually makes brain activity visible. Now the graphic on here shows a PET scan, and it shows a control group uh, and they're not on any cocaine, and then a brain that's on cocaine. You see the difference between the two. That's on the right side of your screen. On the left, that is an EEG cap. It's in a 1010, what's called a 1010 configuration. There's supposed to be the image is not quite correct, but there's 10 on one side and 10 electrodes on the other side of your head, and then we pick up brain waves from that, your brain waves, which range anywhere from 2 or 3 cycles per second, or hertz, up to 125 hertz. Let's look at the basic parts of your brain. Basic parts of your brain. And we're looking at this obviously from the outside. And from the side, this is what's called a lateral view. The front, so we have the frontal lobe here, the frontal lobes here. This lower area here where you see this little protrusion right there, that's called the olfactory bulb. That is the only part of your brain that's almost directly connected to the sense organ itself, your nose. Your temporal lobes, this indentation here, that's called the central sulcus. The primary motor cortex right here, the primary somatosensory cortex. Whenever you see somato, that means body. Sensory is sense. So in here, we associate our motor function, our body function, with our sensory function. This is called the parietal lobe right in here. We'll get into what all of these different areas do eventually. Your occipital lobe, primarily for vision. This back here is your cerebellum. Your cerebellum is responsible for fine motor activity, motor learning, and motor control. When I say motor, I mean your muscles. And then, of course, right on down to your spinal cord. Looking at this from another angle, and also looking now at the interior of your brain, more inside your brain, we're looking at what we call your hippocampus here, right here. These violet curved structures right here. The amygdala on each side, and it's bilateral, which means on both sides. Your brain is bilateral. So there's the amygdala here, amygdala on the other side. This section right here is called the septum. Then these structures that go back like that are your left and right cingulate cortexes. Septum, amygdala, mammillary body. Oh, and the fornix. Now, these structures are below our cerebrum. Our cerebrum was the last area that evolutionarily developed. And humans have the most, and we're still developing it. So, as a segue, what I just said, your cerebral cortex. The cerebral cortex is, in fact, the outer layer of your brain. It contains about 70% of the neurons in your central nervous system. So your cerebral cortex, it looks like just a big, giant, wrinkled walnut. And of it consists of those 2 large hemispheres that cover the upper part of the brain. So there's hemispheres. So here's the right side over here, the right hemisphere, the left hemisphere. And these are divided into 2 lobes, obviously left lobe, right lobe, and that separation leads to what we call corticalization. Corticalization. Corticalization is an increase in size and wrinkling of the cortex. So our superior human intelligence is due, in fact, to this corticalization. But our cerebral hemispheres have and take on different functions. Some of it is subtle, some of it not so much. When you look at the right and left brain functions of our cerebral hemisphere, left hemisphere, right hemisphere. Now, don't get really wrapped up into this, but generally speaking, our left brain is pretty much analytical thought, logic, language, science, math. The right brain is holistic thought. It's spatial function. Diagram. How you think in terms of large, small, square, rectangular, circle. It's part of intuition, creativity, art, and music. So how do we divide our brains? How does hemispheric specialization work? The so-called split brain. And how do they connect to one another? Our brains, our two hemispheres, are connected by a huge trunk of fibers that is called the corpus callosum. So this corpus callosum connects the two hemispheres. So again, here we see that there's this left brain, right brain dichotomy, where the right brain is music and art and dance and perception, fantasy, and the left brain is math and language and science and writing and more sequential types of stuff. Now, what medicine and medical science learned is that if one has very, very severe epilepsy, and I mean really severe, like you're seizing, somebody seizes every 15 minutes, that's very dangerous. And there are a very few cases where the corpus callosum, the individual's corpus callosum, is severed completely. Well, now that's all that does. All that does is that it keeps the left brain and the right brain from communicating with one another, and we get the so-called split brain. The split brain process, where we have learned that is how we've learned that one part of the brain is really better at one thing and the other part of the brain is really better at something else. 95% of humans use their left hemispheres for speaking and writing and understanding. And the left hemisphere is math, judging time and rhythm, coordinating order, analysis, breaking information into parts, and processing information sequentially, that is in order, like 1, 2, 3, 4, 5, one order after one item after the other. This stuff doesn't come automatically, folks. The brain does it for you. In fact, and I'll say this won't be the first time I'll say this, your brain presents your reality. Your brain presents your reality, instant by instant, instant by instant. These words, the image that you're looking at, everything, your sense of it's your brain is presenting that to you. You're not aware of it because it's your brain. That's what you're used to seeing. That's what you're used to being presented. But that is your brain. You have learned to see. If you hadn't learned to see, if your brain hadn't learned to see, even with perfectly good functioning eyes, you couldn't see. You couldn't hear. So on. The right hemisphere is good at recognizing patterns and faces and melodies and expressing and detecting emotions and holistic processing, or in other words, intuition, looking at things in one chunk. As I said, processing information simultaneously, all at one time. All right, let's talk about what these various lobes of our brain do. We'll talk about the frontal lobes first. The frontal lobes control a sense of self, to some degree, motor control, higher mental activities, reasoning, planning, and we call this executive function. Executive, why? Because it's the boss, or at least we hope it is. It also contains the primary motor cortex, and it controls motor movement. Within the frontal lobes, we also have another type of neuron called mirror neurons. Mirror neurons. And they become active when motor action is carried out and when another organism is observed carrying out the same action. It's sort of like when you see somebody yawn and all of a sudden you yawn too. That's part of that. So what else? What do the frontal lobes do? Well, they're located at the front of the cortex. You can see that here. So here's the frontal lobes. They contain association areas, which means that literally that not is association. So when you see something, if you are reminded of something, that reminder is that association. The front of the frontal lobes, that's called the prefrontal cortex, and that's involved in one's sense of self and reasoning and planning. The left frontal lobe contains what we call Broca's area. Now, Broca's area is very important in language, and again, it's 95% of people. It's in your left frontal lobe, and it's in the rear part of your left frontal lobe, but it's still in your left frontal lobe. And it's related to grammar and syntax and pronunciation. So if Broca's area is damaged from either an injury or stroke, a person may know what he wants to say, but can't say the words. So Broca's area or motor or expressive aphasia is what we call that. Any aphasia whatsoever, is a language disturbance resulting from brain damage. You can see here now, this brain, and you can here's the temporal lobe here. These are the frontal lobe out here is Broca's area, and like I said, it's in the rear part of the frontal lobe. The frontal lobe, here's this, and then here's the temporal lobe, auditory cortex, and then this part right in here, that's called Wernicke's area. Right in there, and we're going to talk about what Wernicke's area, because Wernicke's area and Broca's area, there's a very important neural pathway between these two. They have to work in concert, Wernicke's area and Broca's area. Now, the parietal lobes are located on the top of the cortex, and they contain the primary somatosensory cortex. It's a staging area, receiving area for bodily sensations like touch, pain, temperature, and so on. Hence, somato meaning body, sensory. The temporal lobes are on each side of our cortex, and they contain primary auditory information, So that's basically our area by which we hear. Our brain hears. Your ears, they're just instruments sitting out there to carry the vibration to your auditory cortex. It still has to be decoded. Your left temporal lobe contains, again, what I just mentioned earlier, Wernicke's area, and 95% of the people, it's on the left temporal lobe, others 5%. It's related to processing language comprehension. So if it's damaged, the person has problems with meanings of words, not pronunciation. Or in other words, let's say somebody has an intact Broca's area but a damaged Wernicke's area. Oh, they can pronounce words just fine, but it's all nonsense. The occipital lobes are located on the back of your cortex, and they contain your primary visual area. They contain all of your visual association area. They help you to identify objects and faces. Any damage in your occipital lobes can cause what we call agnosias, And a visual agnosia is an ability to identify seen objects. A facial agnosia is the inability to perceive familiar faces. And then we go into what is called your subcortex, and these are the brain structures immediately beneath the cerebral cortex. They're divided into your hindbrain or brainstem, your midbrain, and your forebrain. So your hindbrain includes your medulla. That, as I mentioned much earlier in this lecture, controls vital life functions, your heart rate, swallowing, breathing. In your hindbrain also, or your brainstem, sometimes it's also called, is your pons, is a bridge. And it acts as a bridge between your medulla and other structures, and it influences sleep and arousal. Then there's your cerebellum. Now your cerebellum back here, it almost looks like another little brain in the back here. And it's located also, it's located at the base of your brain. It regulates posture and muscle tone, muscular coordination, and that sort of thing. Your hindbrain also includes what's called the reticular formation. The reticular formation is associated with alertness, attention, reflexes, coughing, sneezing, vomiting. The reticular activating system, or RAS, is it's part of your it's simply part of your brain that keeps you active and alert. The reticular activating system acts like the brain's alarm clock, and it activates and arouses your cerebral cortex. And as you can see here in this diagram, that from the coming up into the brainstem from the spine, we're getting input from touch, pain, temperature receptors, that sort of thing, going up into your spine, and from your eyes, it goes up and it all gets passed onto your brain via your reticular activating system. Your forebrain contains your limbic system, and it's a part within the forebrain closely linked to our emotional responses and our motivating behavior. It provides that primitive quick pathway to arouse your cortex. And within that, we have what's called a very important part of our brain, our forebrain, and that's called the limbic system. We'll get into much more detail about the limbic system as well, but it includes the thalamus, the hypothalamus, the amygdala, and the hippocampus. Hippocampus, very important to memory. The thalamus is a relay system. It's like a router, actually. It's a router for our sensory system. The only thing it doesn't route is olfactory signal, in other words, smell. Hypothalamus regulates emotional behavior. It also is our overall a developmental timer. It tells our bodies when to start puberty. And it tells us just basically how we develop over a period of time. Our amygdala is associated with fear and rage responses. The hippocampus, as I said earlier, is associated with a strong permanent memories, and it also helps us navigate through space. I don't mean outer space, I mean through the space in which we actually occupy. And then we're going to move into the endocrine system. I'm going to take a break here and start a new video, and then we will launch into the endocrine system. I stopped just when we started the endocrine system, and now I'm back. And in fact, I really could have just kept going. We only have a few more slides left. But the endocrine system is a system of glands. They are glands that pour chemicals or hormones, actually, that's what they are, chemicals, hormones are chemicals, directly into the bloodstream or the lymph system. They're carried throughout the body, and they affect many of the internal activities of our bodies. Hormones have a tremendous effect on our bodies, not only instantaneously, or in other words, hormones can tell us to, like you've heard of, all of a sudden I got an adrenaline rush. Well, that's a hormone rush. That's a rush. All of a sudden you have a lot of energy. And the endocrine system has an effect not only instantaneously, like I said, but also it has lifetime effects. It affects, for example, when we go into puberty, when we stop, and so on. It speeds and slows our development through life and changes our development through life. Those glands include the pituitary gland, sometimes called the master gland, the pineal gland, the thyroid gland, and the adrenal glands. The pituitary gland, which is located right here. You see that little sac that's hanging down right there. Here's the brain, the cerebellum behind here, and here's the forebrain here, the prefrontal lobes, right here in here. There, that you see right there, that's the corpus callosum that connects the left brain with the right brain, the brain stem here, and the spinal cord. Okay, well, here's the pituitary gland. It also releases another hormone called oxytocin, which regulates behaviors involved in happiness, pregnancy, parenthood, sexual activity, social bonding, and trust. Sometimes it is even called the social or love hormone. The pituitary gland, as I said, is also sometimes called the master gland because it influences all the other glands, including the thyroid, the adrenals, and the gonads. The pituitary gland, however, has its own master, and that master is a part of the brain called the hypothalamus. What happens if you have pituitary problems? Well, too little growth hormone, which is a hormone that the pituitary gland releases. Too little growth hormones means that a person will be smaller than average. So hypo means under, hypo like a hypodermic needle, hypopituitary dwarfism. So as adults, they're perfectly proportioned, but they're tiny. It is treatable. This condition is treatable by using human or synthetic growth hormone, which will tends to add a few inches. Another type of pituitary problem is called gigantism. So too much growth hormone leads to excessive body growth. And acromegaly is enlargement of the arms, hands, feet, facial bones, and that's also due to too much growth hormone secreted, however, secreted late in the growth period. Then there's the pineal gland. Now, the pineal gland has remained, or had remained, that is, it hasn't, had remained a mystery gland. No one knew exactly what it did. René Descartes, back in the 16th century, believed that it was the seat of the soul. However, the pineal gland regulates body rhythms and sleep cycles, and it releases a hormone called melatonin. Many of you may have heard of melatonin, and that responds to daily variations in light. The thyroid gland in our neck regulates metabolism. So hyper is over. Hyperthyroidism, an overactive thyroid person, tends to be thin, tense, excitable, nervous. And hypothyroidism is an underactive thyroid. The person tends to be inactive, sleepy, slow, obese, and depressed. Then there are the adrenal glands. Now, the adrenals arouse your body and help us to adjust to stress. They regulate salt balance and sexual function. They're located on top of our kidneys. The adrenals themselves are separated into two parts as well. There's two different types of adrenals there. There's the adrenal medulla, that's the core of the adrenals, and the adrenal medulla produces epinephrine or adrenaline. The technical name for it is epinephrine, and that's associated with fear or flight. And norepinephrine, noradrenaline, is linked with anger and fight. Then there's the adrenal cortex, and it produces corticoids. They regulate salt balance, among other functions, but they are corticoid. You've heard of corticosteroids. That's also from the adrenal cortex. So what adrenal malfunctions, what happens? Well, oversecretion of adrenal sex hormones can cause virilism, which are exaggerated male characteristics. For example, a bearded woman. It may also cause premature puberty if oversecretion occurs too early in the person's life. Let's talk about handedness for a minute or two. That is, are you left-handed or right-handed? And that is what handedness is. It is a preference for one's right or left hand. Sightedness is a broader preference for right or left hand, foot, eye, ear. You may notice that some people tend to, when you're going toward in a direction you need to turn, maybe they always go to the right rather than the left or something if there's a choice to be made. We talked also about the dominant hemisphere, the right and left hemisphere, usually produces the side of the human brain that produces language, not necessarily the side controlling the preferred hand, though. And handedness is also partly inherited and also partly shaped by environmental factors as well. Oversecretion of the adrenal hormones, as I said, will cause virilism. Oops, I went backwards. So what are the advantages of right-left hand? Some left-hand preference is due to birth trauma, and that could lead to some health problems. Lefties, the data shows also tend to be a bit more accident-prone, possibly because the world is engineered for righties. Lefties may be more artistically and mathematically inclined due to a more active right hemisphere. And lefties are less lateralized, so they're specialized in abilities of brain hemisphere and hence better survive brain injuries because they could shift hemispheres. They're not so lateralized. Or in other words, there's the difference between the two hemispheres is not so extreme. So that is pretty much what I have covered on chapter 2 on brain and behavior. And thank you very much for